Okay, so I was just telling you about the importance of protein folding, right? So I was talking about protein folding and how complex the process is. So if I just give you an example, like suppose you have a 100 amino acid protein, and if it is doing a random search, right? And there are 3 bonds that could be rotated. So how much time a norm, if it is doing a random search, it would take about 10 power 23 years. That's what we have calculated quickly, right? But a protein folds in a millisecond to microsecond or milliseconds to seconds, and it always folds back to the exactly same structure. That is a specialty of because I guess you know that a lot of functional things in our body are proteins. So a protein is a functional entity of our body. Right? Suppose you just had your food, right? Now if you had a food, what is telling you the taste of the food? It's basically one of the receptor, the molecules that you are taking are binding to the protein receptors, and then that is transmitting to your brain, right? So all the functional aspects, the proteins are called workhorses of a cell, because all the functional aspects are done by the proteins. So it has to come back to the exactly same position or exactly same structure for it to be functional. And I gave you an example. Suppose it doesn't fold back to its functional thing. So you might be surprised that a lot of major diseases, for example, you must have heard of autism, right? Autism is a disease where you basically your brain hand or your coordination goes down, right? So or even other muscular dystrophy and these kind of diseases, you will be surprised to know that a single amino acid mutation, that is by mutating one amino acid to other amino acid in the entire protein structure, because it changes the structure of the protein so much, that is what is resulting in kind of diseases. So in order to either understand a disease or even to develop the drugs, you need to understand the molecular interactions in the proteins. And as I told you, so if a protein is not folded correctly, it is called misfolded state, and misfolded state will result in aggregation of proteins. For example, one of the best example is, now you all eat eggs, right? So what is it that so if you see the normal egg, that is, so the inside it is a liquid, right? But once you heat it, it forms this white, basically you have a yolk inside and then the white protein is outside, right? So what is that? That is nothing but an aggregated state of the protein, right? So the white one that you are seeing is an aggregated state of the protein. In a normal state, it is in a folded form. But once you heat it to like 80 degree, 90 degree, 95 degree, whatever you are boiling it, then the proteins will come together, they'll start misfolding, the structures kind of changes, they'll misfold and they'll start aggregating, and that is what results in aggregation of proteins. So the white part that you see in the egg is misfolded. So similar to that, when in the stress conditions, when your body undergoes a lot of stress, a lot of proteins undergo this kind of aggregation and form these plaques. Okay, plaques means these are fibrils kind of things, and this is what results in a lot of neurodegenerative diseases. And so it is very important to understand the entire process of protein folding. But it is also important to understand the process of protein unfolding as well. So protein folding is something very important. Protein unfolding is also very important, and as I said, so all these are done by your physical methods. So why is protein unfolding important? So when so if you see a cellular compartment, if you see a simple cell, right, there are various compartments in the cell, like there is an extracellular, there is an intracellular part of that. So proteins have to go from one part of the cell to other part of the cell, right? And for going from one part of the, so if suppose in your normal body, or if you, how many of you are aware of the process called diffusion? How many of you are aware of the process called diffusion? What is diffusion? So basically a movement of molecules across a concentration gradient, right? So when there is a concentration gradient, there is a movement of the molecules. So for that, the diffusing region should be larger than the size of your molecule. But in a cell, it is not possible. You have a folded protein which is around 4 nanometer or 5 nanometer. You cannot have so big of pores. So the proteins will actually go from one part of the cell to other part of the cell by basically getting unfolded. So there are enzymes on this, in this region, which kind of unfold the cell, and then they mechanically or they basically energetically they transport the proteins from one part of the cell to other part of the cell. So it is very important process. The protein just as protein folding is important, protein unfolding is also important. Now can you think of ways how we can unfold a protein? So protein folding is a spontaneous process. You prepare a linear amino acid chain and then put it in a solution like in a buffer or in whatever solvent, it immediately folds back, just like I showed you I showed you this part, right? So what is happening? You have your unfolded protein here, and energetically it actually folds back into its native state. Now if I have to move from a native state to the unfolded state, how do you think I can unfold a protein? Well, like what are the methods in which I can unfold a protein? Enzymatic unfolding, yes. There is chemical unfolding. So basically, as I told you, so all this protein structure is held by what? It is mostly held by hydrogen bonds, right? So you need to disrupt these hydrogen bonds, and if you can add a chemical which can break those hydrogen bonds, then it will become a linear polypeptide chain. So there are multiple chemicals that are available, like urea, like guanidine hydrochloride, and these kind of things. So those are chemically, they can unfold the proteins. But you can also unfold the proteins, just as I told you, you can unfold the proteins by heating. If you can actually heat it, increase the temperature, even in those cases, the hydrogen bonds break and it becomes this. But in the body, you think chemical unfolding or thermal unfolding is possible? Can our body unfold the proteins by chemical means? So specificity is the problem. Now, as I said, proteins are all basically held by hydrogen bonds. Now you want to unfold one specific protein, right? You want to unfold one specific protein. Now you cannot change the chemical environment of that particular protein, around that particular protein, because there are other functional proteins also. So when you change the, if you change the pH is not like you are not changing it locally, right? Even the surrounding it also changing. Or if you are adding a chemical moiety to that, even the chemical moiety, because of the concentration gradient, it will diffuse out. So that chemical is accessible to all the proteins. That means other proteins which need not be unfolded will also start unfolding. So that will result in, again as a result, a protein unfolding will result in unnecessary diseases and unnecessary conditions. So if you have to unfold a protein, or you cannot even do a thermal unfolding, you can't heat the body to 90 degrees just to unfold one particular region, right? So both chemical and thermal unfolding is not possible in the cellular environment. So the only way a protein is unfolded in a cellular environment is by using mechanical force. Okay, so there are these enzymes called molecular motors which convert chemical energy to mechanical energy. So just like your fans are what? They are converting your electrical energy into mechanical energy, right? These are kind of piezo's. So you will be surprised to know that these enzymes, these are molecular motors, these are nano enzymes or nano motors. They convert the chemical energy from ATP hydrolysis into mechanical force, and then they kind of unfold the protein and then translocate it. So all the translocations or all the unfolding of the proteins in the cellular environment is happening by mechanical unfolding. Okay, so now before I go into mechanical unfolding of this, so I want to slightly switch gears and talk to you about another interesting topic. Basically, as I told, so you have a thermal unfolding and you have a mechanical unfolding. The problem with the mechanical unfolding is, so if you have a protein and you are changing the chemical environment, if suppose there are 10 different molecules, 10 different proteins of the same kind, and you are basically changing the chemical environment around them, all of them will experience the same change in the chemical environment. Or if you have 10 molecules and you are changing the temperature around it, all the 10 molecules will experience the change in the temperature. But force is not that way. So suppose, I'll give a simple example. Now if I basically put a gas into this room, some chemical gas, all of you would be experiencing, depending on how fast it diffuses, all of you will be experiencing the same chemical environment. Or if I increase the temperature of this room, right, all of you would be experiencing the same. But mechanical force is not that way. Mechanical force is anisotropic nature. If suppose I have to apply a mechanical force on somebody, it is only applicable to that particular region, right? It doesn't get transduced. So that is why, because of the anisotropic nature of the mechanical force, most of the unfolding in the cellular environment happens by mechanical force. So that is something that you should understand, that mechanical force is always applied on single molecules. Okay, now I will switch gears and I will talk to you about some of an advanced topic in biophysical chemistry, which is called single molecule biophysics. Okay, so I guess in the last 40-45 minutes of my talk, I am able to convince you that basically for understanding the biological systems, you need to understand their physical properties. And for understanding the physical properties, you basically look at the various physico-chemical properties, right? Now I'll move on to an advanced topic of biophysical chemistry, which is called single molecule spectroscopy or single molecule biophysics. Let us see what it is, and then I'll move on to that, okay? Okay, so this is an advanced topic of single biophysical chemistry. The initial part I gave you some basics of biophysical chemistry. We will move on to one advanced topic which is called single molecule biophysical chemistry. Now, what is the single molecule biophysical chemistry? Now, you all do spectroscopy. I guess you have gone to NMR spectrometer. You have gone to UV visible spectrometer. You have measured the samples, right? Now, do you measure this? So when you are measuring any spectroscopic reading, are you measuring it on individual molecules or are you measuring it on an ensemble of molecules? You are measuring on an ensemble of molecules, bulk properties. Now, when you are measuring on the bulk properties, the problem is for larger systems like proteins and this. So, just like all of you, how they are different because of the larger system, because these are now proteins have multiple chemical moieties. Even a small variation in the arrangement of the bonds will slightly change its property, right? It will slightly change its function. Now, when you are measuring the bulk property, so if I say that, okay, the class average is 70. That only tells you that, okay, the class is good, but it will not tell how one is doing in comparison with the other, right? What is the heterogeneity in the system that you will not know. So, over the last 20 years, a new field has emerged which is called as single molecule spectroscopy, and it has been mostly used to biomolecules, so it is called single molecule biophysics. Okay, we will talk about that, okay. So, if you look at, this is basically the outline, why should we study single molecules? And there are two methods in single molecules, monitoring methods, manipulation methods, and then what are the kind of biological problems that we are able to address, and then we will see what are the limitations of this. If anybody is interested, these are some of the reference textbooks for the single molecule. This is a very advanced biophysical chemistry topic. The entire field started in probably late 90s or early 2000s, and it is now it's one of the hot topics of biophysical chemistry, okay. Now, if you see this is an example of a bunch of snakes. Now, can you identify, or if I give you this, and if I ask you to draw the shape of a snake, will you be able to draw the shape of any of the, you will not be able to identify what each snake is doing or how each snake is obtained, right? So, basically you will be missing the information of the structure of that, right, of each of this. Similarly, like in this class, I am sure a lot of you are listening, but there will be one guy who is currently sleeping, which I am not able to figure it out, right? For that, I need to do what is called a single molecule biophysics or single molecule spectroscopy. So, the advantage of single molecule spectroscopy is it will give you information on population heterogeneity. Population heterogeneity means how each of the molecule is behaving differently from within the same set. So, under the same conditions, so the conditions are the same, but how each of the molecule is behaving differently from the other. So, that is called as population heterogeneity. Sorry. So, it will give you what is called as stochasticity. Do you know what is stochasticity? Stochasticity means randomness. So, there are a lot of events which are random in nature. Like if you slightly go to a larger and larger system, if you give a simple chemical molecule, if you take a chemical molecule with 5 carbon atoms, you can always predict exactly what is the, if you are doing a reaction, you will be able to predict exactly what the reaction is. Now, if you go to a larger system, because there is so much of variation within the system, there is something called stochasticity, which is called randomness. That means the same molecule. So, the first one is population heterogeneity. That means how each molecule is behaving differently, right? How each of you are behaving differently, right? Stochasticity means the same molecule can behave differently at 2 different times, right? That is called stochasticity, or that is called randomness. So, you are basically giving, the molecule is the same, but it is, and why does this happen? So, if you are taking a simple protein molecule, the solvent environment around it can change, right? The hydrophobic interactions can change. So, basically minor variations around it can result in stochasticity. And it can give what is called as rare events or energetically metastable states. So, if you are basically doing a normal spectroscopy, right? what you get is an average of it. The problem with the average system is if there is an outlier, that means if something is basically different from the average, you will not be. So if I take the class average is 70, but there might be one guy who might not be doing very well in the exam, but he is doing something very interesting other than the studies. So unless you probe each of those persons separately, you will not be able to identify that, right? So any of the rare events or energetically metastable states, that information will be missing when you are doing a bulk study. That for that you want to do what is called a single molecule spectroscopy. And structure and dynamics of individual molecules. As I said, if you want to understand the structure or the dynamics of individual molecules, you need to do single molecule study. Concentration independent mechanisms, right? Because we are doing the concentration, we are doing the measurements on a bulk systems. So all those bulk systems, as you know, the rates of the reactions or anything is dependent on the concentration of the systems. But if you want to understand the concentration independent mechanisms, you need to look at this basically single molecules. And the last one is what I have told you, which is anisotropic properties. So as I told, most of the isotropic properties like temperature, concentration and all those things, a chemical nature, these are isotropic in nature. That means they act similarly on all the molecules. But I gave you an example. For example, I told you, like if a protein has to be unfolded and sent across the cell, you cannot use chemicals to unfold it, or temperature to unfold it, or pH to unfold it. You have to do it mechanically, unfold it, right? So mechanically, when you are unfolding, it is basically an anisotropic property. That means individual molecule, whatever you are doing is only applicable to that particular molecule. It is not applicable to other molecule, and also in the direction in which you are applying. So those are called anisotropic properties. So if you want to study any of these properties, you need to do what is called a single molecule spectroscopy or single molecule studies. So have you, does any one of you, did any one of you hear of single molecule spectroscopy before? So you have heard of spectroscopic techniques, right? But in the spectroscopic techniques, you are only dealing with the bulk studies. As I said, you are not individually dealing. What do you think is a limitation? Why was this field so late to develop? Why do you think people could not understand or realize the importance of single molecules? By the way, I think you will understand it more if I give you a simple example why single molecules are important. Like bulk, we all know bulk studies are important, but if you want to do a single molecule studies, I'll give you one simple example. Now, you have heard, I told you again, DNA. DNA is nothing but a chemical moiety, right? It is basically, now you have heard of something called DNA sequencing. So these days it is so easy that you just take a small blood sample or any part of your thing, you give it, it will give you the entire DNA sequence, right? So it will tell you, it will map on your ancestry, it will tell you what genetic disorders you have, what everything it will tell you about you and your ancestors and like how the information has been passed. Now, if you take a single molecule sequencing, that means basically now each of these DNA strands, so if you give a multiple DNA strands, so each of them will give a different signal. So then the average, you will get an average value of that. So you will not be able to identify how each of them is behaving. But if you actually give them a single molecule, basically if you give a single DNA strand out of here, so it is going to measure or it is going to sequence the DNA strand and it is going to tell you what is the DNA sequence of that, right? So for those kind of things, you only need single molecules. You cannot do it on a bulk studies. You need to have an apparatus or instrument that can measure single molecules. You cannot take your entire blood sample and do a DNA analysis because there will be multiple, that will give you multiple readouts and it will basically you will not be able to identify individual readouts of that. But if you want to identify individual readouts, you need to do what is called a single molecule sequencing, and it is a very common technique these days for DNA sequencing. Right. So to do any of these, basically any of these properties, if you want to understand, you have to go to single molecules. But understanding single molecules is very difficult. And why do you think it is difficult? Why do you think understanding single molecules is difficult? So the problem is the instrumentation limitation. It is nothing to do with so if you are looking at the single molecules, right? If you look at the various time scales, so you're talking about you look at the various length scales. So if you're talking about the radius of a carbon atom, you have around picometer, right? If you are looking at the diameter of DNA, it is around nanometer, and then if you go slightly to the cells and other things, it is around micrometer. So you need to have an apparatus that is actually able to measure this range of nanometers to micrometers very, very accurately. And if you are talking about energetic terms, so if you are for breaking a single CC covalent bond, so basically these are the energy terms that are necessary. Don't worry about the energy terms, but if you are looking at the time scale of this, right? So the time scale has to be very, very low. It has to be in terms of nanoseconds. So you need to have apparatus that are able to measure changes in nanometers and changes in accurately within a time scale of nanoseconds. So that is where the issue was, where people could not develop a lot of instrumentation for doing this. And again, in the last 2 decades or so, this entire field of single molecule biophysics came into picture, because you are easily able to identify the sizes in the range of nanometers within the time scale of nanoseconds. Okay, so your single molecule techniques, for example, how many of you have heard of the term AFM, atomic force microscope? Atomic force microscope. Okay, so atomic force microscope is commonly used to image a very, very small molecules, right? Now, if you are looking at the AFM tip, so basically the cantilever tip is you have basically how it works is you have a cantilever, then there is a tip. So this is called cantilever. Okay, then this is the tip attached to that. So the tip is around 20 nanometers, and if you are, or even you can go to 10 nanometers, and if you go to the protein of the size of the protein, 3 to 4 nanometers. So you cannot measure anything accurately if the size of the probe is larger than the size of the molecule measuring, right? So if you want to measure anything very accurately, your size of the probe has to be smaller than the size of the molecule, otherwise you will have multiple molecules associated with that. So that is why for a long time this entire field of single molecule biophysics could not come out, because they could not measure this size small molecules. And now, because of the high instrumentation, very good instrumentation, we can even measure angstrom level scales within femtosecond time scales. Okay. Now, okay, so if you look at the single molecule methods are basically classified into 2 manners. One is monitoring techniques, the other is manipulation techniques. So, have you heard of fluorescence? How many of you have heard of fluorescence? What is fluorescence? Relaxation of? So when you excite a molecule to a higher energy state, it relaxes from the higher energy state to a lower energy state, especially from a ground vibrational level of S1 state to any of the vibrational level of S0 state in a radiative manner. That is the important one. So fluorescence is a radiative phenomena, right? So that is fluorescence. Now if you want to monitor any of the single molecules, the 2 commonly used methods are called fluorescence spectroscopy, or other is called force spectroscopy. So it is called, the one is called monitoring technique. So in this you label a molecule with a fluorophore. So fluorophore means a molecule that fluoresces. Okay, so you know what is chromophore, right? What is chromophore? Something that absorbs. Fluorophore means something that fluoresces. So if you want to observe a single molecule, for monitoring techniques, you have a molecule, either a molecule or biomolecule, and attach a fluorophore to that, and you monitor basically that fluorophore. That is called monitoring techniques. This is a part of fluorescence technique. The other one is called manipulation technique, where you can actually manipulate a single molecule. So here you are not changing the status of the molecule. So molecule is doing whatever it has to do, the molecule is doing, and you are just observing it by tagging with a fluorophore. Whereas in the other case, you can actually manipulate this, and you can change that by using force. So one is called fluorescence spectroscopy, the other is a force spectroscopy. Now, does the force spectroscopy ring any questions to you? Have you heard of the word force spectroscopy before? Have you heard of the word fluorescence spectroscopy before? Yes. And force spectroscopy? Okay. So generally, if you say spectroscopy, how do you define spectroscopy? Interaction of electromagnetic radiation with the matter, or to be or some people say interaction of light and matter, right? So, but the IUPAC actually kind of not IUPAC. So there is a slight change in the definitions. So interaction of energy with the matter, that is called as spectroscopy now. So because force is not an electromagnetic radiation, right? Force is mechanical energy. It's not an electromagnetic radiation. So when you have interaction of some form of energy with the matter, that is, people are calling it as spectroscopy, and so that is why it is called force spectroscopy, and this is called fluorescence spectroscopy. So fluorescence spectroscopy will help you understand, will help you in monitoring single molecules, whereas force spectroscopy will help you in manipulating the methods. Okay, I just basically whatever I told you is this. So if you want to observe the single molecules, if you want to observe single molecules in near native state, or if you want to track them, like when the structure is changing or the dynamics is happening, if you want to track them, or if you want to label a biomolecule with external probe, so this is where your monitoring techniques are useful, which is your fluorescence. If you want to change the energy landscape of the molecule, right? If you want to change the energy landscape of the molecule, or if you want to apply external force to perturb the system, or if you want to change the study the anisotropic properties, in those cases you use what is called manipulation technique, which is called force spectroscopy. I will show you the tools that are commonly used in these two methods. Okay, the first one is can I hide this? Okay, I'll just put it down. Okay. Okay, so monitoring techniques are basically so monitoring techniques are basically the ones that help you to monitor the process. So some of the commonly used fluorescence spectroscopy methods for monitoring are, it's called fluorescence correlation spectroscopy. Again, as I said, these are some of the advanced topics, because you should be exposed to these advanced topics. This is something that started in probably around 20-25 years back, this entire field, but you will not have them in the textbooks, because most of the textbooks that you are studying are probably 40 years old textbooks, right? So because you should know what is the current status of the research in this field, right? What is the current methods? So fluorescence correlation spectroscopy, so this is called FCS, fluorescence correlation spectroscopy, where you actually, so this is a laser beam, so you basically trap a small volume, a femtoliter volume, and you have basically a molecule that is coming in and out of this particular volume, and this volume is being monitored for its fluorescence. So when a fluorophore comes into this and leaves it, there is a change in the fluorescence, and how fast it comes and how fast it goes depends upon the size of the molecule. So this fluorescence correlation spectroscopy, or FCS, will give you information on the size of the molecules, or if as I told you, if you want to monitor the process of aggregation, or if you want to monitor the sizes of the molecules, single molecules, so this technique is very important, which is called FCS, fluorescence correlation spectroscopy. The other one is called as SM FRET, single molecule FRET. Have you heard of the word FRET? So those of you who have learnt about fluorescence, right? There is another thing called FRET, which is fluorescence resonance energy transfer, or it is also called as Foster resonance energy transfer. Now what happens is, now in your normal fluorescence you have a electron, you are basically shining light, and it goes into excited state, right? Now it is coming back, it comes to the ground vibrational level, and from the ground vibrational level it comes to the ground electronic state. This is called fluorescence. This much you probably know, right? Now this energy gap, whatever is that, this is called as, so when this, so this is basically one of the fluorophore. Let me see if I can draw. So is there a vacuum thing here? There it is. Okay. This is connected right already. So now you have a simple, so you know this is your absorption spectrum. So you have intensity versus wavelength lambda, right? This is your absorption spectrum. Now if you have fluorescence, fluorescence will be blue shifted or red shifted compared to absorption spectrum? Blue shifted, okay. I think, okay, I'll tell you basics of fluorescence then, you very quickly. So this is your, you all know this S0, S1. So this is your electronic state. Okay. Now you have an electron which goes into a higher excited state. Okay. And it comes down to the lower vibrational level. From this, it is coming down as fluorescence. Okay. This is absorption. This is fluorescence. Now the energy of fluorescence will be lower or higher than absorption? Lower. So when energy is lower, wavelength will be higher or lower? So will it be red shifted or blue shifted? Red shifted, right? So basically if this is your absorption, this will be your fluorescence, right? Now if this is your absorption, this is your fluorescence. Now if you have another molecule which can absorb in this region, right? If there is another molecule which can absorb in this, this is absorption of some molecule B, this is the absorption of molecule A, fluorescence of molecule A. Okay. If the molecule is, there is another molecule which can absorb in this region B. So whatever the fluorescence is coming down, it will not come down radiatively. This will be reabsorbed by this molecule B. So that means you will not see the fluorescence from A, because if this molecule was not there, you will see the fluorescence from A. If there is another molecule B which can absorb in this region of this, in this region of the fluorescence, so you will not see fluorescence of A, but what you will see is fluorescence of B. Because the A is coming down, fluorescing, but this energy is reabsorbed by this B, and so that will give fluorescence. So this is called fluorescence B. This is called as energy transfer. So energy transfer from one molecule to other molecule. Okay. So this, when this happens in a fluorescence setup, that is called fluorescence resonance energy transfer. So when the wavelengths of two molecules are matching, so that is called as fluorescence resonance energy transfer. Okay. Now coming back to this. Okay. So this is called fluorescence resonance. So you now, if you have a molecule, so this is a protein molecule that we have been talking about. If you have a protein molecule, now if you attach a fluorophore to one of them, that is called donor fluorophore, and then you have attached another fluorophore which is called acceptor fluorophore. That means the acceptor fluorophore should have an energy absorption in the region of the donor fluorescence. So in that case, what happens is there is a change in the energy, there is a transfer of energy from this donor to acceptor, right? And this energy transfer efficiency depends on how close or how far they are, you see. So basically you see this is, if they are close or far, the energy transfer efficiency will be different. So for a single molecule, you can change that, you can monitor the changes in the conformation. Suppose you have a acceptor here, a donor here, there is an energy transfer. Now if the distance increases, the energy transfer efficiency decreases. So by monitoring the energy transfer of a single molecule, you can guess or you can predict the changes in the conformation of the molecule. So this is called a single molecule FRET, which is another single molecule fluorescence techniques. So similarly, as I told you, one of the first example that I told you, which is basically a DNA sequencing, right? Now you can measure what is called as conductivity of individual DNAs in this. So you have one DNA molecule that is going through that, and each of the bases, so you have only 4 bases in DNA, right? A, T, G, C. Now each of the bases will have different electrical conductivity. Now this, what we are talking about is, these were all fluorescence, right? So in this case, what we are monitoring is conductivity. So each of the base will have different conductivity, and depending on the current that you are observing, you can say whether it is A or T or G or C, and by doing that continuously, you can determine the sequence, the entire sequence of your DNA. As I said, once you know the DNA sequence, you will know everything about a person. Okay, if you want to understand a person, you don't have to either the past or the diseases or anything. You want to understand that, or the physical traits, you can just sequence the DNA and you will get all the information. So for doing those single DNA sequencing, you need to do it at a single molecule level. So in all these methods, we are not manipulating the system, we are just monitoring the system. So these are called single molecule monitoring methods, or basically fluorescence based methods. Any questions on this? Because as I keep saying, this is something that is slightly advanced for your level, but I try to keep it as basic as possible. Any questions here before I move on to the manipulation techniques? Okay, good. So these are monitoring techniques, but there are something called manipulation techniques. Manipulation techniques means, so you are basically, so in the previous case you are not changing anything, you have attached the fluorophore, whatever the molecule is doing you are just monitoring it, right? That is called monitoring technique. Now if you want to change the molecule, if you want to change the energy landscape of a molecule, or if you want to change the pathway of the molecule, you need to use what are called as manipulation techniques. And these are some of the commonly used manipulation techniques: single molecule atomic force microscopy, optical tweezers and magnetic tweezers. So all these are advanced single molecule biophysics techniques which are used to manipulate the single molecules, and you can do a simple molecule or a biomolecule depending on what you want to change. You can do this. So if you see the difference between all of them is, you see the spatial resolution of optical tweezers is between 0.1 to 2 nanometers. We are talking about 0.1 nanometer. 0.1 nanometer means how many angstroms? 0.1 nanometer means how many angstroms? Ten power. So what 1 angstrom is 10 power minus in meters how much? Minus 10. 12 is pico. Minus, right? 10 power minus 10 is what? Your angstrom meters. 10 power minus 9 is nanometers. So 0.1 nanometer is what? 1 angstrom. Can you imagine 1 angstrom? So do you know what is the, if you have studied the hydrogen atom from your things, so what is the distance in the hydrogen atom? Basically from the bond length or whatever you want to call it, or from the nucleus to the outer electron, the distance is around 0.529 angstroms. So we are actually able to monitor to the level of just 2 hydrogen atoms. That means such a precise technique, you are able to monitor or you are able to change the distances to around 0.1 to 2 nanometers in using these methods. And you see the temporal resolution is the temporal resolution in these techniques is slightly high. This is milliseconds, but if you go to the fluorescence methods, the temporal resolution is very, very high. Okay, and if you look at the force ranges, so these techniques can apply force ranges from a 0.1 to 100 piconewtons, and as I said, pico is 10 power minus 12. So they can go from 0.1 piconewton to 10 to the power of 4 piconewtons. So they can have a whole range of forces. Now this is something that you will keep hearing depending on no matter where you go, something called AFM, atomic force microscope, which is a very, very commonly used technique in material science as well as other ways, where you want to, if you want to understand the resolution, or if you want to understand the structural changes at an angstrom level or at an atomic level, you use this instrument called as atomic force microscope. And we can use this technique, atomic force microscope, to apply forces on the molecule or change the forces on the molecules, and then move it to you can change it in piconewtons. You can apply the forces of piconewton and you can monitor the distances of around 0.5 to 1 nanometer. Okay, so this is something that I wanted to quickly tell, the advanced topics that are currently there in biophysical chemistry, and what are the questions that we are asking. Now in the last 5 to 10 minutes that I have, what I want to do is, I want to explain to you the kind of research that goes on in our lab, because till now what I have talked is, in the first 1 hour or 45 minutes I have given you a brief overview of biophysical chemistry, what are the techniques. In the second 40 minutes I have given you some information on one of the advanced topics of biophysical chemistry, which is single molecule biophysics. Last 10 minutes I will give you some information on the kind of projects and the kind of research work that is happening in my lab. Okay. Okay, so when I start the lab, so basically I need to tell that my lab members I should thank, and all the fundings I should thank. Okay, so my lab has a lot of people and it is funded by multiple sources. We will see what kind of, I'll just tell you what is the work that goes on in my lab. Okay. So I think by now you might have understood that our lab is basically a biophysical chemistry lab. We work on biomolecules using physical chemistry techniques and physical methods. So we use methods like, as I said, we use mostly fluorescence and force spectroscopy that I mentioned. We use optical spectroscopy means your UV visible spectroscopy, CD and all those kind of things, right? Optical spectrofluor and that also includes fluorescence and force spectroscopy. We also use some of the biochemical methods like molecular cloning biochemistry for proteins and nucleic acids. We produce a lot of proteins or enzymes in our lab. So just like you synthesize organic molecules, you also have to produce the proteins or enzymes which are much larger in size. So we produce those, and then we do a lot of biochemical assays. Okay, so currently our lab is working on three problems. I'll just talk about it. One is to look at novel drug targets for Mycobacterium tuberculosis. So I guess you all know what is TB, tuberculosis. Tuberculosis is caused by an organism called Mycobacterium tuberculosis. So we are trying to develop novel drug targets. So drugs are different. Drugs are small molecules that are developed. Drug targets means where you want to target. So you are developing small molecule, but that has to go and bind to some protein or enzyme and inhibit its action, right? So we are working on identifying the novel drug targets for TB, and in addition to that, we are also very much interested in looking at some of the fundamental problems. As I said, protein folding, unfolding, protein translocation, protein degradation, these are fundamental problems. Like as I said, they all have applications, but even fundamentally also we do not understand much. So our lab is also working on this. So very briefly, so these are the 4 projects that we work on, and so one is basically we are trying to understand enzymes called mechanoenzymes. As I just told you, mechanoenzymes means something that does catalyze a reaction, right? Now if an enzyme catalyzes a mechanical reaction, that is called mechanoenzyme. That means these are nothing but your nanomotors that convert the chemical energy into mechanical energy, and this has, so there are a lot of motors in our body that move from one part to other. It's not like our body. So when I am talking, there are a lot of cells in our body where molecules are carrying cargo, that means carrying information, not information, cargo from one part of the cell to other part of the cell, or there are proteins that are unfolding, right, enzymes that are unfolding the proteins. All these reactions require a lot of mechanochemistry. So we look at what is called as mechanoenzymes. So we try to understand this process of mechanoenzymes, and then we are looking into another, as I told you, protein folding is a very complicated problem, right? I just told you that you give a sequence and it always folds back to that, and it folds back to it within milliseconds to microseconds to milliseconds, sometimes seconds. Now you can increase the complexity in that protein folding process by adding what is called knot. I guess you all know what is knot, right? Like your shoelace knot or something. Normal polymers, knots are common, but they do not increase the complexity. But in the proteins, because they are always folding back to the same structure and they have to do it very fast, forming or unforming or unknotting is a very complicated process. Okay, so we are trying to understand this process of knotting and unknotting. When we are trying to understand means I am trying to understand the energetics and the kinetics of this process of knotting and unknotting. And the other thing that we are looking at is basically as I said, TB is something that we are working on. So we are trying to address some of the proteins that are helping in cell division of the MTB, and we are trying to arrest that. That means, so any cell has to survive. You all know, in your basic biology you must have studied, for a cell will divide, it will keep dividing, right? You all studied these basic things. Now for cell division to happen, there are some proteins that are responsible for this cell division. So this is basically, you know what is like mitosis or something. So basically if you look at this, there is a cell division that happens, and there are proteins that are responsible for this cell division. Now if you can identify small molecules that can arrest this cell division, the cell will no longer grow, and that will result in the death of the cell. So that can be used as this can be used as a drug target, and you can develop small drugs to that. And then other project that we are working on is called biomolecular condensates. Biomolecular condensates means, so there is a new theory that is coming up. So a lot of reactions happen in the cell, right? Now when as I just told you, when a reaction is happening in the cell, it should not disturb the other compartments or other proteins or other kind of enzyme. It should be very local, right? One way to do it is using mechanical force, but every time you cannot use mechanical force. So there is a recent hypothesis that is going on, is cell forms what are called as biomolecular condensates. So have you heard of phase separation? Have you heard of the word phase separation in your thermodynamics or something? Or immiscible liquids? Immiscible liquids are nothing but phase separate. So if you add water and oil, they kind of phase separate, right? So there is an entire hypothesis that is going on that inside your cells there are biomolecular condensates. Condensates means, so all your cells are nothing but aqueous media, right? So it is nothing, there is no organic solvent there. It is all aqueous media. Now within the aqueous media there are smaller compartments which are membraneless compartment. It means whenever there is a reaction that has to happen, this compartment will form, and once the reaction is over, this compartment will break. So by that what happens is you are doing a reaction in a very localized environment, as well as the concentration is so high that the reaction increases faster. So this is one of the hypotheses that is around 10 years old now, so which is basically called biomolecular condensate, and we are trying to understand what kind of interactions govern this kind of biomolecular interactions. So this is something that we are working on. Okay, I will not talk about much about my research. As I said, if somebody is interested, they can always talk to me, and I'll just stop here, because if you are okay, one thing that I quickly wanted to tell about is knotted proteins. I told you, right? So I do not want to talk too much about these things, but one thing I want to tell about is, you all know that TB is a very serious disease in the tropical countries, right? And many of the Western countries do not work on the TB because it is not a disease for them. It is only a disease for the tropical regions. So we have to come up with a solution for that. Now, as I told you in the beginning, how do you develop a drug? You need to first identify certain targets. Targets means you need to identify certain proteins that are very different from our proteins. Like if you are developing a drug that inhibits both our proteins and MTB proteins, then it is of no use because it is also going to inhibit us. So you need to identify certain targets which will basically inhibit only those the proteins of the particular organism, and for that you basically and then develop these drugs. Okay, so here are some of the enzymes that are these days that are being used, which are basically like proteasomes, and so don't worry about this particular I think that's a recorded thing. Okay, so basically there are enzymes called foldases, proteases, proteasomes, unfoldase that we are targeting to understand this. Okay, so I will not talk much about this. You can ask me at a later stage if you are interested. Okay, and similarly, this is the protein which is called FTSZ, which helps in division of the cell. So this is the cell division. So cell is dividing. So this is the protein. This forms a nice polymers like this. You see, this is a TEM image, transmission electron micrography image. So these are nothing but protein fibrils. Okay, so these fibrils are the ones that are essential for this dividing of the cell. Now if you can develop a small molecule that can inhibit this, that can break these polymers, then you are going to disturb the cell division. By disturbing the cell division, you can actually design a drug target because you are not allowing this molecule to do that. So we have come up with some of the molecules. For example, this is one of the chemistratin kind of molecule which can inhibit the polymerization of this. So this is something that we are working on. Okay, so again, that is very briefly what we work on. We work on like a lot of mainly two problems. One is MTB related enzymes and the other is protein folding related questions. Okay, so with this I'll stop here, and if you have any questions, because it's 5 to 3. If you have any questions, if you want to understand, because I know that this is some topic, this is topic that you have not been exposed to before, but this is very much part of the chemistry. And if you go to any bigger institutes like or even abroad, biophysical chemistry or chemical biology is a hardcore chemistry subject with an interdisciplinary nature of both physical chemistry and biology. Okay, or physical chemistry, organic chemistry and biology. Yeah. So any questions you would like to ask, you can go ahead. Okay, so I think I was also told to tell about what are the opportunities that you have for these things. I think by now you would have understood. It has a huge applications in pharmaceutical sectors, like in the entire area, the entire industry of drug discovery or entire pharmaceutical industry depends upon identifying the drug targets and doing proper biophysical assays to that. So biophysical chemistry has a huge applications in pharmaceutical industries. And even if you are looking at if you can understand this, people are also using these principles for understanding the degradation of biomaterials. For example, I didn't tell you much about it, but one of the more pressing problems these days for biophysical chemists is modifying the activity of certain enzymes that can degrade plastics. So in the last 5 years, if you have seen, there are some new enzymes that have been developed which can degrade your plastics, right? This is nothing to do with drug discovery. This has more. So you are in chemistry, we are more interested in making the molecules, right? But you should also think of breaking those molecules. So you all know how big a problem plastic is, right? Now any plastic needs to be degraded, and any of the other harsh, biologically it is not possible to degrade plastics. But now there are enzymes that have been able to chew up, or that have been able to degrade PET, you know PET, right? PET, yes. PET or even nylon kind of plastics. On those kind of plastics, the enzymes are there. So we are currently monitoring or modifying the amino acid sequences so that you increase the enzymatic activity, so that it will actually increase the degradation rates. So that is for the sustainability part of it, but for large applications is in the pharmaceutical sectors. Okay. So if there are no questions, then we will stop it here. Okay.